Hi everyone, welcome back. Today we're going over learning and memory, which is absolutely one of my favorite things to think about and to talk about in the context of behavioral neuroscience. My PhD was essentially in learning and memory and using preclinical models of substance use disorder to interrogate learning and memory. So this lecture and then the lecture on substance use disorder will be fairly well, I mean, it's not going to be like super dense, but I'm going to talk about it because this is, I enjoy talking about it. So I'll try to be clear about what it is that you need to know, though, and not get too far into the weeds. But we have, this is the first lecture of Module 4, so we have learning memory, we have neuropsychiatric diseases, disorders, and substance use disorders. So the last three of the class. So hopefully you guys will also enjoy this and be glad to be done with biopsych pretty soon. So let's first define learning and memory. Learning, is a change in behavior due to prior experience. It's acquiring new information or a change in behavior due to practice, study, or experience. So those are basically the two definitions of learning and memory. At a fundamental level, a learned event is one that changes behavior due to prior experience. Memory is the ability to retain information and to retrieve that information. So the change in experience or the change in behavior part of learning kind of insinuates a memory component. So we have, this kid studying for a spelling bee, and then we have the kid performing at the spelling bee, right? Obviously, this being the learning component and this being the memory or the ability to recall information component. Memory is something that is kind of inherently controversial because it has to do with consciousness. So at least most of the memory and most of the learning that we care about has to do with consciousness. As you'll see, there are many forms of learning or many types of learning that is not conscious. But I think a lot of the things that we, when I initially talk about learning and memory, you're probably thinking about the concept of, thinking about something that happened to you in the past, right? Thinking about the exam, thinking about the date, thinking about when you first met that person, when you first experienced ice cream, whatever the case is. Like I think that tends to be what we think about when we think about memory. But there's actually a lot that goes into these basic types of learning and memory. So no doubt you have heard of Ian Pavlov and B.F. Skinner, as these names are extremely fundamental to the study of psychology. And they are essentially the fathers of classical conditioning, Ian Pavlov, and operant conditioning for B.F. Skinner. So you'll need to know that Ian Pavlov is the guy that coined classical conditioning or associative learning. You'll need to know that B.F. Skinner is the one that did operant conditioning. Ian Pavlov is kind of an interesting character because he was a physiologist. He studied eating behaviors. He studied the stomach and the gut, and he didn't really want to be known as a psychologist. He was a physiologist, and this was his main thing that he was interested in. And if you remember from the methods in behavioral neuroscience, as well as our lecture on eating and hunger and feeding and all that, I kind of alluded to two ways that people tend to get to behavioral neuroscience, and that's either from the psychology perspective or from the physiology perspective. So when you have people that are interested in physiology, it ultimately will lead to psychological research because physiology and things that affect physiology are going to have inherently psychological aspects to them, or things that have to do with the brain, which will eventually be some kind of behavioral effect. Similarly with psychology, people that come from a psychological realm will have to learn about the physiology because you can't have behavior without physiology either, right? So anyway, this is an apparatus that Ian Pavlov produced to study classical conditioning. Okay, so classical conditioning is the pairing of an unconditioned stimulus with a neutral stimulus to form a conditioned response. So let's go ahead and unpack some of these terms. An unconditioned stimulus is an external stimulus that produces an unconditioned response. So what do I mean by stimulus? Anything. An unconditioned stimulus is something that's out in the world that when you experience it, it produces some kind of unconditioned response. Unconditioned in this situation means unlearned, okay? So if you take your hand and you put it on a hot stove, you will reflexively rip your hand off of the stove, right? That is an unconditioned response. You don't need to learn that the stove hurts, because as soon as you put your hand on the stove, you will, without thinking, remove your hand from the stove, assuming that you have, a non-pathological nociception system. So long as the pain signal can get to your brain, you'll remove your hand. Another example will be fear. If you have something that's threatening your life, and you have a fear response to that. Now you can learn that some things are scary, you know what I mean? But if you, there are certain things that you will not need to learn. I'm hesitating in what I'm saying because, not everybody's afraid of spiders and snakes, but there is some evidence that people are inherently kind of afraid of spiders and snakes and things like that, but not everybody is. So I'm hesitating a little bit to say that. But a fear response is not necessarily a learned response. If you get hurt, if you see something that is threatening, you will have a feeling in your gut of fear or anxiety. That would be an unconditioned response. And if it is to something that you have never associated with an outcome that's negative, like a spider, like many people have never been bitten by spiders, but they're still afraid of them. Then this is an unconditioned stimulus. The spider being the unconditioned stimulus, the fear of the spider being the unconditioned response. And so that brings us to this definition here. It's a physiological, a UCR, or an unconditioned response is a physiological or behavioral outcome that is unlearned and is a reaction to the unconditioned stimulus. So again, you having anxiety over when you are stepping, looking over a cliff, when you are running, and you essentially react by running away or by screaming. I'm a very jumpy person, so if you sneak up on me, I might yell at you. That's kind of an unconditioned response that I have to a startle, like some kind of event that was startling to me. Now, a neutral stimulus is an external stimulus that does not produce any particular outcome prior to learning. So a neutral stimulus will be, maybe these keys. There's nothing special about these keys. You see them, you heard them hit the table. It didn't elicit any kind of response to you. They are unconditioned. They are unlearned. They're a neutral. They're not unconditioned. I'm sorry. They are just a neutral thing that's out in the world that has no effect on you, and you haven't learned anything about it. Then we have a conditioned stimulus. This is something that was formerly a neutral stimulus that has been paired frequently enough with an unconditioned response to associate the neutral stimulus and the unconditioned response, now making the neutral stimulus a conditioned stimulus, okay? So when you have and we're going to go over this with specific detail with what Ian Pavlov discovered, but the point of this is that when you have a neutral stimulus, if you associate that, or if you put it in close temporal or temporal proximity, which means close in time, with a unconditioned response or an unconditioned stimulus, then the neutral stimulus will start to produce the previously unconditioned response. When that happens, it becomes a conditioned response. And so that's what we have down here. A conditioned response is a learned response that occurs after presentation of the conditioned stimulus, okay? So if no learning has occurred, and when you see the thing that's in the universe, and you have some reaction to it, that's all unconditioned, unlearned. right? I see the spider. I've never interacted with a spider before, but I feel fear, okay? In that scenario, the unconditioned stimulus is the spider, and the unconditioned response is the fear. If I burn myself and I move my hand away from the stove, the unconditioned stimulus is the hot thing, and the unconditioned response is the reaction to me moving my hand. Now, let's say that I'm looking at the coils on the oven and they're not on, right? And I burned myself on those coils yesterday. And then when I look at them today, I have a feeling in my stomach of, either fear, anxiety, or frustration, or anger, or maybe I even physically react to seeing the coils. That is a conditioned response. Because it previously, now, and also, like, that's like a little bit confusing because, and it also exhibits kind of the nuance here, which is that the coils themselves, there's nothing wrong with the coils. The coils don't actually burn you. It's only when the oven is on and the coils are hot that you can get burned, right? So if you have coils on your stove and they're not on and you reflexively jump away from them, you now have a conditioned stimulus with a conditioned response, even though the initial thing, which was the hot coils and you removing your hand away from it, was an unconditioned stimulus with an unconditioned response, right? So this can get a little bit tricky depending on how we're wording things and the timeline of what we're talking about, right? Because initially it may be the case that you see the coils on the stove, you touch them when it's not on, and it doesn't elicit anything because the coils are not hot. Okay. You could also have the scenario where you're not afraid of the spider, right? So the spider could be a neutral stimulus because maybe you aren't afraid of spiders. But then you get bit by the spider, right? That biting event causes pain and causes an unconditioned response, which is you yelling or screaming or moving your hand or whatever. And the unconditioned stimulus in that scenario was the pain from the bite. Now, let's say that after that scenario, you see spiders and now you're afraid of spiders. Now spiders have become a conditioned stimulus that have a conditioned response, which is when you see the spider, now you're afraid. But it's not because you were always afraid of spiders. It was because you were afraid of the spider biting you. You learned that the spider is harmful. So let's go ahead and take a look at what Pavlov specifically discovered, or how he discovered these things. Holy moly, I'm already kind of like 10 minutes in. So I need to hurry up. In any case, so what Pavlov was doing, he was studying the digestion. He was studying the eating behavior of dogs. And so every day they would go in and they would feed the dogs. And they noticed that when the graduate student or the research assistant, whoever it was walking in, that the dogs were already salivating, okay? And they were curious as to why the dogs were salivating before they had eaten. So this is a normal response. Salivation in the response to food is normal, right? So we have the dog is going to eat, and that is going to cause saliva production, right? So this is an unconditioned stimulus, the food that is presumably in the bowl. The bowl itself is a neutral stimulus, right? But it will be paired with the food, so it will become a conditioned stimulus. But in this scenario, let's just pretend this isn't a bowl, it's food, okay? So the food is an unconditioned stimulus, and when you put the food in the dog's mouth, then saliva will start to be produced. This is an unconditioned response because it's just a physiological outcome. There's no learning that needs to happen. It's just the case that when you put food or kibble or whatever in the dog's mouth, then saliva is produced. It's a response to the stimulus, and it's unlearned. Now, what was going on in Pavlov's lab is that there was, as the story goes anyway, there was a bell that was on the door. And whenever the graduate or the research assistant was coming in to feed the dogs, the bell would make a sound, obviously, because it's on the door. And that neutral stimulus, which was the sound of the bell, became associated with when the dogs were going to be eating. So this turned that neutral stimulus of a bell into a conditioned stimulus, which caused the dogs to salivate, which is a conditioned response, right? So in the beginning, you can see that when we have something that is an unlearned phenomenon out in the world, such as salivation in response to food, these are unlearned things. We have an unconditioned stimulus, which is the food, and an unconditioned response, which is salivating. You can take something that's neutral, that has no effect, like a bell or a sound or a tone. You can ring the bell enough times in close proximity with the food, and now the dogs start to salivate. And this is true not just of animals. I mean, this is true with humans. If you think about Sour Patch Kids, maybe you really like Sour Patch Kids and Warheads and sour candy, and you're starting to salivate just literally thinking about watermelon or strawberry Sour Patch. I think the strawberry is my favorite, the strawberry Sour Patch. Maybe you don't like Sour Patch, maybe you like chocolate and you're thinking of Reese's, or you're thinking of Hershey's or Kit Kats or Snickers, and maybe you start salivating thinking about that. People will have varying levels of sensitivity to this, but I guarantee that eventually we could talk about something that's going to cause you to have a physiological reaction. So associative learning is something that is very real in humans, as I'm going to give you some examples of. So this is actually something that happened to me in grad school. Let's go ahead and just read the example. I develop a habit of vaping after work in my car. I park my car in a parking structure. Vaping too much makes me nauseous. One day, as I'm walking to the parking structure, I notice I start getting nauseous as soon as I see the structure. Now, this literally happened to me in grad school. I was vaping a lot, and I was getting too addicted to the vaping. I was like doing it in the bathroom, in the lab, whatever. And so I started leaving my vape in my car so I wouldn't go hit it in the bathroom, like in between experiments or whatever I was doing during the actual workday, because my car was like literally a mile walk away from the lab. So, I went to University of Florida, and the parking there is just awful. Like at UB, we're so lucky that we have the parking that we do, because parking at UF was absolutely nightmare. But in any case, I would leave it in the car because it was like a freaking 10-15 minute walk away from the lab, so that would pretty much be enough of a deterrent for me to not go get the vape. But the point is that I was parking in this structure, like a big parking structure, and I started getting very sensitized to the nauseating effects of nicotine, right? So if you remember from our psychopharmacology lecture, you can become sensitized and tolerant to different effects of the drugs, right? So I was doing nicotine, and I was becoming sensitized or overly sensitive to the nauseating effects of nicotine. So I would like hit the vape, and then like after like one or two vapes, I'd get like so nauseous. But in any case, when I was walking towards the structure, I would see the structure and I would start to feel nauseous just by looking at the structure. This is very clearly an example of associative learning or classical conditioning. And this is also what happens to individuals when they're receiving chemotherapy, and they get what's called a conditioned taste aversion. So typically when people are getting chemotherapy, they may intentionally eat a what's called like a icicle of a particular flavor that they don't care about, to associate the nausea that they get from the chemotherapy with that particular flavor of that icicle. So then that way they don't become nauseated in close proximity to the food that they eat when they get home, because nausea is a super strong learning stimulus. This is why in this particular instance, I literally had to quit vaping because I would get so nauseous just by looking at the parking structure. So anyways, let's see if you can figure out what all the various, what is the unconditioned stimulus, what's the unconditioned response, the neutral stimulus, the conditioned stimulus, and then the conditioned response in this scenario. Go ahead and pause the video or whatever and see if you can figure out what all of the different components are. So the unconditioned stimulus is nicotine. The unconditioned response is nausea, right? So although the nausea was something that happened, I became sensitized to it. This wasn't a learned effect. This was sensitivity of my physiology to the drug. So it is something that didn't happen immediately, but it is an effect that happens from nicotine. So nicotine is the unconditioned stimulus. An effect of nicotine is nausea, which I felt nauseous from the nicotine. The neutral stimulus was the parking structure, but it became the conditioned stimulus because when I saw the parking structure, I would then have the conditioned response, which is nausea. Okay, so the neutral stimulus will always become the conditioned stimulus, assuming that that's how the question is phrased, right? So you should be able to pick out these things. Now, nicotine isn't explicitly marked here, so you could have said vaping for the unconditioned stimulus instead of nicotine, but I mean, like, the reason why I'm getting nauseated from the vaping is because of the nicotine. So let's go ahead and take a look here. A mouse is put into a cage and receives a mild electric shock that scares it. When mice are scared in this way, they freeze momentarily. A high-pitched tone is played during the shock. Subsequently, the tone is played without the shock and the mouse freezes. So see if you can figure out what all the various components are. I'll give you like 5 seconds to pause it. Okay, so the unconditioned stimulus is the shock. The mouse does not need to learn that the shock is going to scare it. The unconditioned response is the freeze. Because when mice are scared in this way, they will just stop moving. And this is actually called contextual fear conditioning. So if you are interested in behavioral neuroscience, this is a technique that is used very frequently where they have mice in a little cage and they give them a little shock. It's not like they're not getting like electrocuted, but it's enough of a shock to scare them and they will have this characteristic freeze response. Anyway, so the neutral stimulus is the high-pitched tone, and the conditioned stimulus is the high-pitched tone, right? So like I said, the neutral stimulus will always essentially become the conditioned stimulus unless the question is trying to trick you. Or if they're referring to a neutral stimulus that's not relevant, right? So you could have a high-pitched tone and then like some light in the other room, whatever. But the conditioned response is the freeze at the presentation of the tone. So this is where it can get a little bit nuanced, right? Because what if they said the mouse freezes in response to the shock? That's an unconditioned response. That's not a conditioned response. If they said the mouse freezes at presentation of the tone, which indicates the shock, then that would be a conditioned response. And that's because that requires a learning component. The mouse is not afraid of the tone until it is in close temporal proximity to the shock, which is the unconditioned stimulus. So other names of associative learning are Pavlovian learning, because Ian Pavlov is the one that discovered it. Classical conditioning or associative learning. These things are all essentially the same phenomenon. So if on an exam I were to say, which is an example of classical conditioning, and I gave you the mouse example, then that would be accurate. If I had said the mouse example and said Pavlovian learning or associative learning, all of these things are referring to the same phenomenon. So don't get tripped up with the different words that people use. They all mean the same thing. Okay, so let's talk about operant conditioning. Now, this is a very young B.F. Skinner here. If you look him up, he has most of his pictures are very old, but I found this one when he was a young man, and I thought that it looked kind of interesting to have this old photo here of this guy from a long time ago. But operant conditioning, defined, is increasing or decreasing a behavior dependent on a task or an external stimulus, i.e., behavior is shaped by outcomes. So that is a very important distinction between operant conditioning or Skinnerian learning, which is different from Pavlovian learning or classical conditioning, which is that the behavior is shaped by the outcome of the learning procedure. These Pavlovian, associative, classical conditioning type things, these are all largely unconscious responses to a stimulus, right? So it's not like you are trying, like if you're someone that's really jumpy and you see a bee and you now have this like phobia of bees and you just like react by screaming when you see this bee. It's not like you're not deciding to scream because it makes the bee go away, right? It's like the behavior is a result of kind of these things that you don't really have too much control over, like the salivating to the mention of a watermelon Sour Patch. Um, whereas operant conditioning is actually a conscious thing. So here we have a picture of what's called a Skinner box. And again, I don't know why all these old scientists are so like not into how they were ultimately remembered by. But he also, as far as I know, did not like the fact that these were called Skinner boxes. They can also be called operant boxes, and that's probably the most technically true term, but a lot of people will refer to this as a Skinner box, and a Skinner box is basically, or an operant box, or an operant chamber, all the same thing, is referring to some kind of apparatus that has a behavioral either punishment, reinforcement procedure within it. So here we can see a rat is in this box. It has a lever, and it can push the lever and potentially get water dispensed, or it could push the lever and get food dispensed. And then there's also an electric grid on here, which may shock the rat, and we'll get into why we would do that. Also, there's a light here, and that's because operant conditioning more or less is a higher order form of learning from associative learning. So when you have operant conditioning or some kind of operant learning response, it is going to inherently integrate associative learning. Okay, so associative learning, like I said, is this unconditioned stimulus turning a neutral stimulus into a conditioned stimulus, and then you eventually getting that conditioned response, right? You can help animals and humans learn by pairing various stimuli with the behavioral response that we're talking about here, so that when you press the lever, the light turns on and then a food pellet comes out. That will help the rat learn a lot faster than having no cue associated with the behavioral response. In this case, the light is the cue. The levers are often retractable, so it's like, here's the wall, the lever will come out, the lever will like come out of the wall, and then the rat can start pressing the lever. That lever coming out of the wall is also sometimes a cue that the rat can start getting, reinforced or rewarded. But anyway, this is what an operant box is, a Skinner box. And we need to know these four terms when it comes to operant conditioning. We have reinforcement, punishment, positive, and negative. Reinforcement means increasing behavior. So you could have positive reinforcement or you could have negative reinforcement. We already talked about some of this in our homeostasis lecture when we were talking about, why homeostatic behaviors are regulated or how they're regulated. A lot of times it's through negative reinforcement. Reinforcement itself just means increasing behavior in this context. Punishment in this context means decreasing behavior. And similarly, you could have positive punishment or negative punishment. And that's not referring to whether or not punishment is good or bad. It is just saying you either have a positive form of trying to decrease behavior or a negative form of trying to decrease behavior. So positive in this context means adding a task or a stimulus, whereas negative means removing a task or a stimulus. So let's see if I have so basically, I think I have some examples here, but I will just go ahead and briefly talk about it. So if I have positive reinforcement, that means that when the rat presses the lever, I want it to press the lever, let's say, then I might, dispense some food to increase the rat's lever pressing, right? The reinforcement is increasing behavior, right? And the positive aspect of this is that I'm adding food to the behavior that I care about. So as we already defined, operant conditioning is increasing or decreasing a behavior dependent on a task or an external stimulus. The behavior is shaped by the outcome. So if I want the rat to press the lever, I give it food when it presses the lever, right? That means positive reinforcement. I could also have positive punishment, right? Let's say I don't want the rat to press the lever. So every time it presses the lever, it actually gets an electric shock, right? Like it's a little bit barbaric, but this is just what it is, right? So if you're adding the electric shock, right, and you want the rat to stop pressing the lever, so you shock it whenever it presses the lever, then this is positive punishment because you are decreasing the behavior by adding the electric shock. So negative reinforcement is removing a task or a stimulus to increase behavior. So this is a little bit maybe counterintuitive, but basically when you have some kind of, basically what this is referring to is when you have a painful or annoying stimulus that if you do the behavior, it goes away, right? So when you're thirsty, it's very uncomfortable to be thirsty. And so drinking removes or alleviates that thirst, right? That's why thirst behavior and drinking tend to be thought of as negative reinforcement, because it gets you to you drink because of the fact that you're in pain or that you're uncomfortable. Now, there are other reasons why humans drink. You know, we drink caloric, we drink like sweet beverages or diet beverages like Diet 7-Up or Diet Coke, or you know, just regular Coke, or you know, we drink alcohol. So humans drink liquids for a variety of reasons. But when we're talking about dehydration, and why somebody will drink in response to dehydration, that is negative reinforcement because thirst is uncomfortable. If we have negative punishment, this is removing a task and decreasing a behavior. So taking away a driver's license after someone has had multiple traffic infractions is an example of negative punishment, because we're taking away a privilege and we're trying to decrease an outcome. So yeah, I already gave this example. Positive reinforcement is a rat pressing a lever for a food pellet. Negative reinforcement is a rat pressing a lever to turn off the electric grid, for instance. So like, let's say that's equivalent to the thirst example. You know, if the electric grid is just on all the time, then you know you could train the rat to like keep their hand on the lever, or their paw, to stop the electricity from flowing. We have positive punishment, giving a student detention when they have outbursts in class. Negative punishment, taking away a driver's license, you know, which is a privilege after someone has had multiple traffic infractions. You know, you have maybe like the thing in your car that is super annoying when you won't fasten your seatbelt, the beep that goes on. That's a form of negative reinforcement, because, you know, they're going to annoy you until you know, fasten your seatbelt. Spanking is oftentimes a form of positive punishment. Not good. You know, hitting your kids is not good, but spanking is a form of positive punishment. You're hitting your kid, trying to get them to stop doing something. I guess you could also try to hit them to increase some kind of behavior, but generally I think it's thought of as a punishment in the sense of you're trying to decrease some kind of behavioral output. So just like with Pavlovian learning or classical conditioning or associative learning, there's various names for operant conditioning, which is Skinnerian learning, instrumental conditioning, and operant conditioning. So I think the Skinnerian learning and the operant conditioning are pretty obvious because those are in the name, but the instrumental conditioning also refers to the same thing. So instrumental conditioning is operant conditioning, and both of those are Skinnerian learning. Although most people don't really refer to Skinnerian learning, most of the time they will say operant conditioning or instrumental conditioning. So operant conditioning, I kind of already alluded to this, often includes classical conditioning. So here's a picture of a rat that's in an operant box, and this is an example of the levers that they will push. And they will push the lever and a light will come on. And pairing a stimulus with a behavioral response that you want to increase will really help teach the thing that you're trying to teach. Often when teaching rodents to self-administer food, drugs, conspecific, so this means Obviously in my PhD, what I primarily did was I taught rats to self-administer cocaine. Self-administer means that they're giving themselves the drug. But they will also do that with food or conspecifics. Conspecifics just means like another rodent. You can teach them to press a lever, and then like a little door will open and allow them to play with another rat for some period of time. They will do that. The light is paired with the reinforcer, and the light itself becomes a reinforcer. Yeah, so this is very interesting, and it's particularly true with drugs. So when a rat is self-administering cocaine or heroin or whatever drug, that is an intravenous infusion oftentimes. So they will press this lever, the light will come on, and then they will get an intravenous infusion of cocaine or heroin or whatever. What happens here is that although this is operant conditioning, because you are doing a form of positive reinforcement, meaning that you want the rat to press the lever more, and so you're giving them cocaine to incentivize that, right? The rat is actually pressing the lever to get the injection of cocaine, and the light comes on. The light becomes a conditioned stimulus, and they will actually press the lever for the light for a long period of time, and actually have like increases in dopamine, for instance, without actually getting an injection of drug. And this is true of humans as well. Many people who, I probably will talk about this in the drug addiction or the substance use disorder lecture, but many people who do have an addiction or some kind of substance use disorder will report that, there's a sense of anxiety that washes over people that are addicted to drugs or have an addiction. And that anxiety, when they're trying to get drugs, or when they don't have drugs, or when they're thinking about getting drugs, or when they're thinking about like running out of their drugs, there will be an immense sense of anxiety, a lump in their throat, like all sorts of feelings of panic and things like that associated with it. And they will report that when they're on their way to their connections house, that their anxiety will already start to alleviate. When they actually get the drug, when they get the bottle, maybe they're alcoholic and they get the bottle of alcohol, or they're addicted to, whatever drug, weed or Coke, or, it could be anything, Xanax. Once they actually have the drug in hand, then the anxiety starts to be alleviated, so just the cues that are associated with the drugs themselves will become extremely rewarding in a sense. And by rewarding, I mean reinforcing. So that is kind of the learning aspect. Now let's move on to memory. Memory is the ability to retain information and retrieve that information. So we have many, this is just the picture I showed at the beginning. We have many different types of memory. So we have short-term memory and we have long-term memory. So first, we'll talk about short-term memory, which is broken up into sensory, working, and non-classified short-term memory. Sensory memory is like when you smell roses or anything, and you can you take the stimulus away and you can kind of remember briefly what it smelled like in a real salient way. Okay, so I'm not saying that like right now think about how Sour Patch Kids taste and you start to salivate thinking about it, right? I'm saying that there is a unique characteristic of particular sensory events that you have like a short-term memory about, like how somebody smells. Maybe you give a loved one a hug and they walk away, and like you can kind of like really still smell them or whatever that smell was for a short period of time. Or maybe you're looking at somebody's facial expression and you close your eyes and you can really picture what that looks like in a real way that is distinct from when I'm saying picture somebody frowning. Or how an ice cube felt when it was on your forearm. Maybe you put you touch the ice to your forearm, you take it away. And you can kind of imagine in a very, very salient way what it felt like when the ice cube was on your skin. You also have working memory, which is learning and using a fact or piece of information. So, remember to cut the red wire instead of the blue wire. So you have the movie scene, the guy is trying to disarm the bomb, and there's a red wire, blue wire, green wire, whatever, and the person on the phone says, "Oh, cut the green wire," and then like all of a sudden the voice is gone, and they have to remember that information and act on it. So that's working memory. Or also, I have this down in non-classified, but you could also put this in working memory. So if I were to say, "Oh, remember the digits 5, 6, 7, 8, 9," because you're going to need to put that into the door to get into the animal facility or whatever. That could be a example of working memory. You also have short-term or non-classified, which would just be memorizing some random facts, like a list of numbers that is not associated with some obvious task that you're going to need to use it for. So if I just said, remember blue, green, yellow, red, green. You might be able to remember that. I mean, I've already forgotten it, but you could, if I said, just do your best to remember these three words: cat, house, shenanigans. You could remember those three things: cat, house, shenanigans, for some period of time, and it's being essentially held in a short-term memory capacity, that will eventually be forgotten. So that's short-term memory. So we also have long-term memory, which is defined by explicit and implicit memory. Implicit memory is procedural, so that's like being able to ride a bike, being able to do a backflip, like having the kind of ability to automatically do something like tie your shoes, that kind of a situation. Whereas explicit memory, which is also called declarative memory, is further separated into episodic and semantic memory. So episodic memory, let's just say explicit memory, first of all, is something that you can say, right? So declarative means that it's something that you could speak out loud. You could say, I took biopsych with Bo in the summer of 2025, and you could be thinking of sitting in this class listening to me lecture about this right now. That would be an explicit memory, and specifically an episodic memory. I always remember episodic memory as in like the episodes of our lives. That's how I always kind of identified it when I was an undergrad. But episodic memory is a personal memory. It's declarative because you could speak about it. And by speak, I don't mean actually verbalize it, although you could, assuming that you can verbalize. But if you are someone who can't speak or whatever, you still have episodic memory. It's just that it would be thoughts instead of, actual vocalizations. But the point is that declarative memory is something that you could actually, talk about in some way. And an episodic memory would be a personal memory. So you remember going to Disneyland when you were 7. You remember sitting in this class listening to me lecture. Semantic memory are facts that are not specific to your personal situation. So you could say, Bo taught biopsych in summer of 2025. That would mean, that's a fact, but you're not saying anything about your personal involvement with that. You don't remember it, and maybe you don't remember. Maybe it's a fact that you did take the class, but it wasn't that memorable for you, so you don't actually remember any actual lectures. You just know that it was a class that you took. You could also say that, Disneyland is a theme park. That is, in Southern California. That is a semantic memory. But you could also have an episodic memory of Disneyland. You could say, well, when I was 7, we went to Anaheim, California, and we visited Disneyland. And I saw Mickey Mouse, and I remember how scared I was, but then he gave me a hug and a sucker, and everything was dope. You know what I mean? So that could be an episodic memory. Episodic memories are personal to yourselves, the episodes of your lives. Semantic memories are factual. They just are truth, things about the world that you could also say. Okay, so you'll definitely need to know the different types of long-term and short-term memory and be able to pick out examples. So we'll get into the neurobiology of memory now, which we won't have a whole lot of time to get through because it can get quite complex. But a lot of times when you're thinking about memory, the hippocampus will probably be the answer. I think I remember said something about, whenever you're talking about homeostasis, it seems like the hypothalamus is the answer. Whenever you're talking about memory, the hippocampus is the shallow answer. It's found in the medial temporal lobe. Hippocampus, I think, like has something to do with Latin and seahorses because it's shaped like a seahorse. It's involved in storing and encoding learned experiences, and it encodes both context and emotional memories. Context meaning the actual environment where you're at. And we found out a lot about the hippocampus and the fact that it's so important for learning and memory because of HM. So you may have heard of HM, Henry Molaison. I'm not exactly sure how to say his name, Molaison. HM, I've always just known him as HM because when I first learned about him, he wasn't dead yet, which is horrifying because he died in 2008, which meant that I learned about him like forever ago. But in any case, HM was a very famous patient in psychology, just like Phineas Gage, where he had a horrific seizure disorder. And so they thought that they could remove part of his temporal lobe to allow him to not have these seizures. But what ended up happening is that they basically just removed his hippocampus, which did cure the majority of his seizures, but unfortunately, he couldn't form new episodic memory. He could develop some limited semantic memory, but new episodic memory was essentially lost to him. So he had what's called anterograde amnesia. And you'll need to know the difference between antro and retrograde. But anterograde amnesia is a loss of memories after an injury or some kind of thing that is causing you to lose your memory happens. Retrograde is when you lose memories for events before an injury. So retrograde is actually kind of more common because when you get a concussion or when you have a severe head trauma, it's very common to have brief retrograde amnesia where you can't remember the moments preceding the actual injury. Anterograde amnesia can be pretty horrific, as we can see here with HM, where literally he couldn't make new memories. So he didn't know that he was aging. He'd look in the mirror and be like, "Dude, what happened to me?" Because like he can't. It's like 50 First Dates, right? It's anterograde amnesia. It's he can't form new memories. Retrograde amnesia, I'm sure that it happens where it's like you just completely forget everything about your past, but I think that that's pretty rare in comparison to like acute retrograde amnesia where the individual, like I said, just loses the time just prior to the accident. So the hippocampus, like I said, is important for both contextual and emotional information, and there are two subregions of the hippocampus, which are the dorsal and the ventral hippocampus. And the dorsal hippocampus is important for contextual learning, while the ventral hippocampus is important for emotional learning. And so what happens here is that this was a slide from a lecture I gave a long time ago about substance use disorder, but basically when you're doing drugs, you have an emotional reaction to the drugs, which is like, okay, this feels good. And then you also have to learn information about where you got the drugs at, which is the environment or the context. So the dorsal hippocampus oftentimes is associated with a contextual or an environmental learning event, whereas the ventral hippocampus is associated with an emotionally salient event. Okay, so you need to be able to delineate those two things. But like I said, the hippocampus is necessary for contextual learning, and contextual learning is a form of associative learning, right? So this is important because this is also saying that the hippocampus is necessary for associative learning. Now, the hippocampus is not the only brain region that's important for learning, though. The prefrontal cortex is also extremely important for learning, and it's found in the frontal lobe. It's the last brain region to fully develop. It is what helps give you executive control over actions, helps you with decision making, and as we're pointing out, is very important for learning and memory on a basic level. So essentially what happens is that there is a circuit between the hippocampus and the prefrontal cortex where temporary information is stored in the hippocampus, sent to the cortex, and then essentially there's a loop, a positive feedback loop that strengthens those memories. So you'll need to know that the hippocampus is very important for learning and memory. You'll need to know that the prefrontal cortex is important for learning memory, but specifically you need to know that there is a positive feedback loop between the prefrontal cortex and the hippocampus that helps you encode learned outcomes. And this is happening in real time when you're encoding information. So now realistically, I'm not going to ask you about this on the exam, but just so you know, it is very likely the case that your entire brain is very important for learned associations and for memory. As much of your brain is contributing to your ability to perceive, have sensation and perception events. And also there's different types, like fear learning and things like this are important in the amygdala. So it's not just so simple to say that it's only the hippocampus or only the prefrontal cortex that's important for memory. The whole brain is likely important, but this just kind of goes to show why learning and memory is such a hot topic, is because it's very complicated, just like the topic of consciousness is a very complicated thing. So let's go ahead and get into some of the mechanistic views of learning and memory outside of just the broad swaths of brain regions that are important. There's also cellular mechanisms of learning and memory. So these guys here, this is a bit of a deep dive. You probably will not have heard of these guys in a lot of your other biopsych courses, but this is what I did my dissertation on, so I like to talk about them. But Richard Semon, he was a zoologist and a physician in the late 1800s and early 1900s, and he gave us this horribly difficult to define definition of the physical substrate of memory that says the enduring, though primarily latent, modification in the irritable substance produced by a stimulus. So whatever that means, basically this is that defined, which is that he said that the physical thing that memory is called the engram. And this is the lasting physical change in the brain state and structure that occur in response to an event or an experience. Because again, what we're talking about is that learning, a learned phenomenon, right, is a change in behavior due to experience. So how do we reconcile that on a physiological level? Richard Semon thought, well, it's probably that there's some kind of physical change in your brain that is happening, and that physical change or that physical change of the pattern of activity of your brain is what the memory is on a biological level. Because before he started thinking about this, people were primarily dualists, right? So everybody was basically running around assuming that memory was just, and consciousness in general, was something that was associated with the soul. So that's why this is so important, because we might take this for granted right now, but in the late 1800s, everybody was still very, very religious, and so everybody thought that the soul was essentially where your memory was housed. And so he was pretty radical about this. He was pretty, he was castigated. He was made fun of a lot in the scientific community because he didn't have a clear mechanism to point to, and everybody was dualist, and so they're like, you idiot, like it's just all in the soul, like what are you even thinking about? He actually had a really hard life and had a very tragic death, but anyway, Carl Ashley is the next guy that we care about, and he said, the trick is not to find where the trace is located, referring to the memory trace. He was building upon Richard Semon's work, but where it is not. And essentially, Carl Lashley was a very pessimistic scientist, and he spent his whole life trying to find where, a memory would be housed in the brain. And so he would teach rats to solve a maze, and then he would lesion various aspects of the cortex of the rat to try and kill the memory. But what he found was it's like no matter where he lesioned, the rat would still be able to solve the maze after learning about it. So what he kind of gave us in the neurobiology of learning is that memory is not localized to a single brain region. It's kind of all over the place, which is kind of what I was alluding to earlier when I said it's not just the hippocampus that's important for memory. And then we have Donald Hebb. And this is something that you can't have a biopsych course without hearing, but neurons that fire together wire together. So this was originally, Donald Hebb never said this, but this is basically, this concept comes from Donald Hebb. Donald Hebb was a student of Carl Lashley. And while Carl Lashley was extremely pessimistic about being able to uncover the biological foundations of learning and memory, Donald Hebb was a theoretical neuroscientist, and although he did have some practice, like some actual experimental data for him, he primarily was a theoreticist. So he gave us the term cell assembly, and he hypothesized that essentially what happens is that we have a group of reciprocally interconnected cells that are simultaneously active during an event. So he hypothesized that essentially we have groups of neurons that when they're activated in synchronicity, or essentially when they happen to be activated together, this causes them to be synaptically connected. Okay, so that when you activate one of those cells, you get the activity of another cell. And so that is what he hypothesized was essentially the biological foundation of memory, is that these neurons that are being activated simultaneously become synaptically wired together, and it's that pattern of activity now that is causing the conscious memory experience. So essentially, if we have, Baskin Robbins, I don't even know if it's here on the East Coast, I'm from the West Coast, so we have Baskin Robbins all over the place, which is 31 flavors, it's an ice cream place, and they have marketing, right? So you see the Baskin Robbins sign, and you get your Baskin Robbins ice cream. This makes you happy because it's delicious, and you make an association between these things, right? So you have some neurons here that are going to be sensitive to just your ability to see the Baskin Robbins sign, right? So you have to have the ability to see the sign. You have to have the neurons that are sensitive to the sugar molecules or the coldness of the ice cream. And then, you have to like the ice cream, right? So you enjoy the flavor. And all of these neurons that are associated with these various aspects of this memory will become wired together so that when you see the Baskin Robbins sign, it makes you think of the ice cream or the taste, or it makes you think of the happiness. But the point is that the memory is actually the activity or the pattern of activity in this group of neurons that we will call a neuronal ensemble, okay? So this will for sure be a test question, which is that neuronal ensembles are essentially the cell assembly that Donald Hebb had alluded to. So Donald Hebb said cell assembly, but what we call that today are neuronal ensembles, okay? A neuronal ensemble is a group of neurons or a subset of neurons that become synaptically connected after repeated experiences. This pattern of activity is hypothesized to be one of the cellular mechanisms of memory, okay? And so I will give a shameless plug of a paper that I published last year, which was in addiction biology. And essentially what we showed here is that there are subsets of neurons within the nucleus accumbens core that encode cocaine seeking. And if you destroy those neurons, you'll get a significant reduction in active lever responses in these rats. So essentially what we did was we targeted the neurons that were encoding the rat's memory of pressing the lever for cocaine. We destroyed those, and then we saw a reduction in cocaine seeking. So this is a pretty hot topic in the neuroscience and behavioral neuroscience of learning and memory, and so a lot of people are now studying neuronal ensembles. So it's not even in the textbook yet. It's fresh off the presses. Anyway, the next thing that we need to talk about when it comes to the cellular mechanisms of learning and memory is long-term potentiation and long-term depression. By the way, you won't need to know this for the exam, but you will need to know what neuronal ensembles are. And I'm just showing you this paper just to kind of demonstrate the actual real-world testing that's going on today to look at these hypotheses. Anyway, so we have long-term potentiation, which is also LTP, and this is a persistent strengthening of synapses. And we have long-term depression, which is a persistent weakening of synapses. So when individuals, electrophysiologists are studying the action potentials of neurons after a learned event, they can actually measure the excitatory postsynaptic currents that are happening. And when LTP is observed, you can see that this excitatory current is increasing. And you can also see that initially, you can see that the excitatory postsynaptic currents here are lower than they are over here. So this increase strengthening in the excitatory signal is long-term potentiation, and this is the kind of canonical view of what's happening in the brain to cause a learned response. We have the ensemble theory, which is the idea that these neurons are becoming essentially patterns, producing a pattern of activity. And that pattern of activity is the actual memory on a biological level. But also, in order for that to happen, we need long-term potentiation to actually wire those neurons together. And long-term depression is just the opposite of that. So long-term potentiation is dependent on AMPA and NMDA receptors, and you should remember from our previous lectures that AMPA and NMDA receptors are glutamate receptors, specifically ionotropic glutamate receptors. So, and if you remember, the Not My Dumbass can remember which NMDA receptor, which ionotropic receptor is the complicated one. We have this being exhibited again here. So NMDA receptors are blocked by a magnesium ion. So we did go over this already in the synapse lecture, but I'll kind of give us a refresher here. But the NMDA receptor has a blockage here of a magnesium ion, right? It's blocking the channel. So when there's an action potential, that allows the efflux of this magnesium ion, which will then allow the influx of sodium and calcium. This influx of cations produces upregulation of more AMPA receptors, okay, which then kind of causes the cycle to continue. So essentially, this increasing AMPA receptor expression is what is explaining this increase of excitability that we're witnessing during long-term potentiation. So we have, an excitatory signal that is sent. We have the activity of AMPA receptors. The AMPA receptor increases the charge of the membrane. That then kicks out this magnesium ion from these NMDA receptors, which then allows the influx of sodium and calcium, when glutamate is around, which then causes the upregulation and the increased excitability of the neuron by increasing the amount of AMPA receptors. Because if we have more AMPA receptors, what that means is that we have more opportunities for glutamate to bind to them, which then gives us more opportunities for the influx of sodium. Okay, and remember, if we have an influx of sodium, that's going to result in an increase of neuronal activity, or excitement at least, of neurons. So here we have, again, what's happening here is LTP induction, which is essentially an upregulation of AMPA receptors. Okay, so that's what this is showing us here, is that we have activity here. This is going to, NMDA receptors are activated. This then upregulates AMPA receptors and causes increased neuronal excitability. But we also have long-term depression, and that's necessary because if you have too much glutamatergic stimulation, you'll get neuronal cell death. So it is a constant balancing act between having not enough stimulation or too much stimulation that these neurons are trying to balance here. So long-term depression is exactly the opposite of long-term potentiation, in that it is the downregulation of AMPA receptors, which ultimately will cause a decrease in synaptic strength. Okay, so that is the end of our learning and memory lecture. You'll definitely need to know, that LTP is the increase of excitability through an increase of AMPA receptors. You'll need to know that LTD is a decrease in neuronal excitability due to a decrease in AMPA receptors. You'll need to know about neuronal ensembles, that it's the cellular basis of memory, and that it's basically the idea that neurons that are synaptically connected and have a particular pattern of activity is the cellular basis for memory. You'll need to know, I ask you about Donald Hebb, Carl Ashley, and Richard Seaman. We'll look out for the study guide so I can determine. I don't know. It depends on how many questions I get out of this because there's a lot of content here. So whether or not I'll definitely ask about Donald Hebb. Let's go ahead and say it there. I'll ask about Donald Hebb. You won't need to know about Lashley and Seaman. Although you should learn about Richard Seaman because he was a very good scientist, very ahead of his time, and had a very tragic end. You'll need to know about the dorsal hippocampus and the ventral hippocampus. The dorsal hippocampus is important for context or an environmental memory, whereas the ventral hippocampus is an emotional memory area. You'll need to know about HM. You'll need to know about long-term memory, short-term memory, and the various sub categories of those. You'll need to know about operant conditioning and the fact that it involves classical conditioning. You'll need to know what negative and positive punishment are, what negative and positive reinforcement are, and obviously you'll need to know about associative learning because we spent, I think, the most time on associative learning. So, be on the lookout for examples like this and for you to be able to determine what the UCS, UCR, and et cetera are. And yeah, that's learning memory. So I know a lot of you were asking for the little summary slides at the end. I stopped making those, I think, after psychopharmacology just because there was so many bullet points at the end of the lectures that I was basically just telling you to know the entire PowerPoint, which I kind of may have just done right now. But that's why they're no longer around, is because I was like, well, I don't really think it's going to help anybody actually prioritize their studying. So I'm going to try and get it's I don't even know what the date is. It's the 7th. So I'm recording the other 2 lectures today, and I'll edit them this weekend, and then hopefully have all the study materials and the videos up for you by Sunday or Monday at the latest, so you can do well in this last exam. All right, I will see you in the next lecture.